Hello, everyone. My name's Eli, and I'm a graduate student in Lakhar Bintu's lab at Stanford. And first, I just want to thank the organizers for always putting on such an excellent event every year, and also for giving me the opportunity to tell you a little bit about our work. I also want to highlight that everything I'm going to talk about today was co-led by another graduate student in the lab, CC Andrews, and it's definitely been a team effort working on this. Okay. The overall goal of our lab is to better understand human gene regulation, specifically through this perspective of transcription factors, which are proteins that regulate the expression of other proteins. And in doing so, determine the gene expression programs that are critical in really all aspects of life, from patterning tissues and directing cell fate choices during development, like Hox transcription factors, or enabling cells to respond to a variety of environmental inputs, such as interferon response factors. Transcription factors are modular proteins. They contain DNA binding domains that recognize and bind to specific DNA sequence motifs, and also effector domains that recruit various cofactors to actually influence the transcription of target genes. And classically, it's been thought that effector domains come in kind of two main flavors: activation domains that recruit co-activators to activate transcription, and repression domains that recruit co-repressors to repress transcription. And a lot of great work, including a lot of quantitative and biophysical modeling, has focused on trying to better understand these domains. And while the question is, for a given domain, how much, when, and where transcription occurs? And while the details are nuanced, we've arrived at a relatively simple canonical understanding of these domains. Typically, for an activator, the more an activator you have and the longer it's bound, the more transcription occurs, and vice versa for a repressor. The more of a repressor you have and the longer the repressor is bound, the less transcription you get. Our lab studies effector domains using tools from synthetic biology. We take potential domains of interest and we fuse them to the bacterial DNA binding domain rTEDR that binds tightly to TEDO sequences in the presence of DOX. We express these synthetic transcription factors in human cells containing a genomically integrated reporter gene. And we use this approach because it allows us to remove a lot of the confounding variables and complexity that is present in the genome, such as the presence of other transcription factors binding and co-regulating target genes. It also gives us a great degree of control over all of the system, or the variables and parameters in our system, which lends itself well to quantitative measurements and modeling. For example, if we include the minimal promoter, min-CMV, in our reporter gene, we can quantify an effector domain's activation strength, and then it's very easy for us to swap this out for the constitutively active PEF promoter that allows us to quantify an effector domain's repression strength. And some other members of our lab have recently used a high-throughput version of this assay to systematically identify and characterize activation and repression domains across the 1,600 human transcription factors. But what was surprising from these screens is that they revealed the presence of bifunctional domains, the same small region of protein that is able to both activate a minimal promoter and repress a constitutively active promoter. And as an example, we can consider argfx, and all of the data that I'm going to show you today comes from this transcription factor, but the principles we think we found have been general so far to other domains we've tested. After 1 day of recruitment of the bifunctional domain to argfx to the minimal promoter, it's able to activate the reporter in around 70% of cells, so a pretty strong activator. However, at 5 days of recruitment at the constitutively active promoter, the same domain is able to silence almost 100% of cells, so it's able to perform both functions. But what's even more interesting is if we look more closely at the gene expression dynamics at the minimal promoter, while argfx initially activates the cells, by 6 days of recruitment, almost all of these initially active cells have turned back off. So we see a transient pulse of gene expression despite constant transcription factor recruitment. And if we recruit these bifunctional domains to medium strength promoters that kind of start in the middle, by 6 days of recruitment, we can actually see a single domain bifurcate the cell population into a group of active cells and a group of repressed cells. And this really runs contrary to our classical understanding of activation and repression domains. And so we wanted to better understand how it's possible for a single domain to dynamically activate and repress the same target gene with constant input. And also, for a given gene, what are the parameters that control the strengths of activation and repression? One observation we had about these gene expression dynamics is that while the fraction of active cells changes a lot over time, the relative or the average expression level of our reporter gene in these cells is relatively constant. This suggested to us that bifunctional domains are possibly creating and catalyzing transitions between discrete gene expression states, possibly through simultaneous interactions with both co-activators and co-repressors that are able to dynamically regulate the chromatin state of the reporter. And we can represent this with a simple three-state kinetic model of gene expression that's similar to those previously used to describe epigenetic regulation. And if the rate of activation is fast compared to the rate of repression in this model, it would predict a pulse of gene expression with constant transcription factor input. So we wanted to test if bifunctional domains actually recruit both co-activators and co-repressors to the reporter. And to test this, we added chemical inhibitors of various cofactors during ArgFX recruitment. We observed that when we added a co-repressor inhibitor, ArgFX only activated the reporter. It completely lost the ability to repress. On the other hand, when we added a small molecule degrader of a co-activator, we impaired ArgFX's ability to activate, but really left its repression strength unchanged. So this told us that these domains are in fact recruiting both co-activators and co-repressors to separately influence activation and repression. We also wanted to measure the chromatin landscape of the reporter over time during bifunctional domain recruitment, and we did this using cut and run. We first looked at a canonically repressive histone modification, H3K9 trimethylation, and observed that it gradually increased over the duration of recruitment. On the other hand, when we looked at a canonically active histone modification, H3 panacetylation, we saw that it actually sort of spiked in abundance initially during recruitment and then gradually decreased over time. And so these domains are able to dynamically regulate the chromatin state of the reporter, and also they have different kinetics of activation and repression, which is sort of consistent with our three-state model. However, one sort of obvious prediction from this model is that for a long enough recruitment time, all of the cells should eventually transition into the silent state. When we extended our time course, however, we saw that a small but sizable fraction of cells actually remained in the active state. And so we needed to return to our models and update them in order to better understand this phenomenon. And we reasoned that there were kind of two versions of our model that could explain or account for this non-zero steady state of active cells: a dynamic equilibrium between the active and silent cells, or the presence of an additional stably active state that is unable to be silenced by the co-repressor. To distinguish between these models, we allowed the cell population to reach steady state during ArgFX recruitment and then sorted the active and silent cells and kept growing them in the presence of DOX. And we actually observed that these populations do not re-equilibrate at all. So these domains are creating long-term stable gene expression states, or kind of terminal gene expression states, with constant recruitment. We also wondered how other parameters, such as domain concentration, would affect these gene expression dynamics and the steady state fraction of cells in the active state. And in our system, we can sort of control the relative domain abundance by changing the amount of DOX we use to recruit the domain to our reporter. As shown previously, at a high concentration of DOX, we see this strong pulse of gene expression with constant recruitment. But to our surprise, when we decreased the DOX concentration, ArgFX only activated the reporter. And by filling in the rest of these concentrations, we can see that ArgFX acts only as an activator up until a certain concentration threshold, past which the rate of repression begins to continuously increase. And this leads to a non-monotonic relationship between the relative domain, or the sort of effective domain concentration, and the final fraction of cells in the active state, which once again sort of is at odds with our classical understanding of activators and repressors. We hypothesized that the relative strength of activation and repression is actually being controlled by the occupancy of the domain at the DNA, or the regulatory region of our reporter, based on some recent observations in the literature. And so to test this more directly, we sequentially removed binding sites from our reporter gene and saw very consistent results. At low numbers of binding sites, ArgFX acted only as an activator and then continuously increased the rate of repression as we increased DNA occupancy. Because these results were so consistent, we wondered if we could create an integrated model that could take both domain concentration and the number of binding sites as input, and then faithfully recapitulate the gene expression dynamics as output. And I don't have time to go into the details now, but we essentially were able to combine a thermodynamic binding model with our kinetic state-based model of bifunctional regulation, and then globally fit this model to both data sets with reasonable success. And please find Cece and I afterwards if you're interested in the details, or stop by our poster. But in summary, we think that through the combination of experiments and modeling that I've shown today, we're beginning to arrive at a general framework for understanding bifunctional gene regulation, which kind of doesn't align with our classical understanding of activators and repressors. Essentially, we think that a variety of inputs, such as the number of transcription factor binding sites, the affinity of these sites, or the concentration of the transcription factor, work together to determine DNA occupancy of the transcription factor, which then determines the relative strength of activation and repression. And by combining different levels of DNA occupancy with different basal expression levels of target genes, a single bifunctional transcription factor can access a rich and flexible behavior space in individual cells, acting as a pure activator, a pure repressor, or in the right conditions, even a pulse generator. And as proof of principle for this framework, we've constructed cell lines containing 3 separate reporter genes that we've engineered to occupy different regions of this behavior space, and shown that a single bifunctional domain can differentially regulate the gene expression dynamics of these 3 reporters in a predictable way. And by changing a global variable in this system, such as reducing transcription factor concentration, we can cause these reporters to lie in a different region of this behavior space and change the resulting gene expression dynamics in a predictable way. With that, I'd like to thank all members of the Bintu lab, especially Cece and Lakra, as well as all of you for listening, and I'd be happy to take any questions.
We have time for a few questions. Yes.
Yeah, hi, that was very interesting. I didn't fully understand how is it that increasing DNA occupancy causes a change from a activating to the potential for repressive behavior. Is the idea that if you have like many TFs bound at one location, they can like in a multivalent way recruit a repressor or something like that? How does that work?
Yeah, so we don't know exact mechanisms, but the idea and sort of what's predicted from our modeling is that activation is kind of more efficient at lower occupancy, like maybe its interaction strength with the co-activator is much stronger. And then you need kind of more copy numbers of domains in order to effectively either recruit co-repressors or allow them to function effectively, maybe through multivalent or cooperative sort of recruitment of the co-repressor complex. But we don't know exactly, but that's what we're thinking.
So the data that you presented remind me a lot of stories I've heard from people working on developing safe harbor sites for cell engineering, where there are issues with long-term silencing of genes that they would like to have expressed. And so I actually have 2 sort of related questions. One is, for the long-term silencing that you see, do you still need docs for that silencing to be maintained? And secondly, have you studied at all the effect of sort of extrinsic factors like locus positioning, the effect of what's the word I'm looking for? Like CTCF binding sites in neighboring regions, that sort of thing.
Yeah, so you're bringing up the question of background silencing, which is like a huge issue in genetic engineering. And we don't think that this is background silencing because it is dox dependent. At the active promoter, you can release dox or wash it out and it will turn back on. And I didn't have time to talk about it today, but we can compare these bifunctional domains to sort of classic activation domains. I think a couple times I showed some data from VP48, which is like a really strong activator. And we do see slight background silencing that's kind of normal for these types of experiments, but nothing like the degree of silencing with the bifunctional domains. In terms of sort of genomic context affecting these variables. We haven't directly tested that. We do all of our experiments at the AAVS1 locus, which is like kind of a well-characterized safe harbor locus. And so by swapping out promoters and changing some of the variables, we like think, or in our like sort of framework, we think that the genomic context would be important for these dynamics if it affects occupancy in any way, which it most likely, or like 100% of the time will do. But we haven't actually like directly tested that by putting it in like other safe harbor loci, for example.